First of all, Chris, thank you for inviting me to give a talk here today. And correctly, I'm from Forum, that you can see here in the picture. It's been there since early 80s and is the base for animal science research at Aarhus University now. We have changed from being a sector research institute to being part of Aarhus University. What I intend to share with you today is outlined here. I'll be talking a little bit about physiological imbalance because I think that's important, but also try to give a status on where are we today in dairy production and how do we see the future. And then I would like to go into on a small bit on what we've been working on, and that is the interaction between nutrition and immune function, and also give some ideas on how we potentially should be preventing diseases through nutrition, but also actually management, and what I see as the future challenges. We have for many, many years, as been mentioned already, been addressing breeding. Does this one work? Been dealing with breeding and feeding and management, and also changing housing and environment for the animals due to changes in herd sizes, etc. And as has already been mentioned, we've obtained very big increases in milk yield and thereby in efficiency, but probably little has happened, if any, improvements in health, and certainly we know that reproduction has become poor. The question is then, what are the consequences going to be in the future, where we are going to use genomic selection? We've had increases in milk yield of up to close to 1% a year. In the future, probably we'll see an increase at 1.5% at a fraction of the cost what it is of the breeding today. And certainly, this whole new area of genomic selection is going to put big emphasis on also improvement in these areas, because as genomic selection still is, as the selection we have nowadays, basically a black box operation. I've tried to look at what has actually happened in Denmark on some diseases. I do not recommend anybody to, particularly not students, to go in and do such comparisons over years because it's basically impossible. I've done it anyway. Because it's impossible because the way of registering diseases has changed, both who does the registration and also the whole management systems around it. So it's not actually doable, but basically, if we go to the bottom line, there has been a slight decrease. I would argue, though, that this decrease is because there are some diseases lacking nowadays because cost has increased. We do not recognize all the ketosis cases that are there because that's just handled in management. So my argument would actually be we have not improved diseases. There might be some differences over which diseases are most important. That primarily has to do with changes in production systems. Many of the diseases we have is in the early lactation from adapting to the onset of of lactation, the calving, and the early part of the lactation period. There we have a very high disease incidence. It has been argued that that had to do with milk yield. But that's really not the case, because milk yield is not high until out here. It's not high in here. It's having an increasing phase in that particular period. So what is it? These are the lactation curves from first, second, and third lactation cows. And we tried to look at it in a different manner, and here you see the acceleration in milk yield. And if we go back to this, that actually starts looking some similar. An acceleration, that is actually the increase per day in milk yield. So it's the adaptation that's basically the problem. And we argue that the rate, that it's probably not milk yield per se, that's the key problem. It's the rate of increase in milk yield. It's due to adaptational problems and what I would call physiological imbalance. And what I'll try to address is a lot on physiological imbalance here in the rest of the talk. So, what is physiological imbalance? Well, there I disagree with you, Chris, in your nice presentation on the film we saw earlier this morning, because we actually are some that have thought that we should improve immune function and health by reducing physiological imbalance in the cow, and then that will at the same time actually improve production and reproduction when the cow is in balance. It's at least 10 years ago that we did that. And how do we then define physiological imbalance? I must say that's really a very broad and not easy to use term. But what we do is that we say, what we define it as is cows whose parameters, and that could be glucose, glutamine, beta-hydroxybutyrate, NEFA, that deviate from the normal. And then we need to find out what's actually normal at a different stage of lactation. But also that the consequences is an increased risk of developing diseases. And that may be clinical or subclinical. And returning to the former slide on disease, that is basically primarily clinical diseases. And my argument would be that our major focus should be on the subclinical. That's just difficult to address because it's subclinical. Thereby, it's not shown directly clinically on the cow, for instance. And also that it reduces reproduction and/or production. But haven't we done that all along? No, we've primarily had a focus on groups of cows, on herds. And this is from an experiment where cows had a diet with a certain energy content, and this is then first, second, and third lactation cows. And what farmers and advisors has been dealing with is how to manipulate that on a herd level in order for to improve health, production, and reproduction. And if we then go in and do that, we can see that we can certainly shift these figures. And if we have cows enough per treatment, we can also show this is significant. But is that really what we are looking for? Is it really important when we're talking about diseases on individual cows? I don't think so. The reason is, and here we have beta-hydroxybutyrate. There's close to 11,000 blood samples here taken across 617 lactations. with different breeds, and that's the basis for these. My argument is that yes, we can see these small changes due to nutrition, but it's not going to help those cows that are in difficulties, because look at these variations that we have. My key point is we need to be focused on these top ones. They are probably the ones that are having the problem. I could make the same figures for NIFA, for glucose, and a number of other parameters, but this is just to show the point. What we have then done is using random regression analysis so that we can actually take each individual cow out. So there's 617 lines here for each individual cow that shows the deviation, the random deviation for that particular cow. My point is that this is not random variation. I did the analysis of comparing milk yield, random regression parameters for each individual cow in milk yield, and then tried to explain that by metabolites, hormones, and energy intake. That could explain 24, 25, and 26% of the variation, and in total, if I included them all, about 50%. So basically, there's a lot of information in the random, what we consider random. It's just random because we don't know what we're dealing with. We have then tried to work on an index for physiological imbalance. The reason is actually that energy balance is difficult to use. It's been used for decades, but it's really difficult to measure in practical circumstances. So we tried to go in and do an index where we used NIFA, beta-hydroxybutyrate, and glucose in a model, and then try to relate that to energy balance and some blood parameters. And we've looked at the correlations. The philosophy is that an index of more parameters probably is better in describing a situation for the cow than just one individual parameter. And this is just the correlations. Clearly, when it's been linked to energy balance, it's not surprising that it's very high for NIFA, but it's also actually high for glucose here. The correlation is just negative. And for beta-hydroxybutyrate, it goes down and up. The key thing is also that what's important changes over time. We've then tried also another difficult thing, that is to relate these parameters to disease incidence. And here again, it's one experiment. It's a big experiment, but it's still one experiment, one environmental setting. But we found that for mastitis, PI, physiological imbalance, and NIFA came out significantly. Those that had mastitis compared to those not had also higher physiological imbalance. That was actually prepartal, because later on in lactation we actually couldn't see any clear messages to the parameters. That doesn't mean I don't think that they are still there and that we should use it to look at physiological imbalance in the animal. Similar for metritis. I've chosen these because it has to do with infections, and there's a lot of variation in immune function, and there's a lot of potential factors going from the genetics, the gender, and the early part of life, but also the physiological state, hormones. We know hormones has a huge impact. Different hormones have impact on immunological factors, but certainly also nutrition, physiological status, body condition, and environmental stress, including pathogens, do influence immunology of the animal. More important is also that there's an interplay between all these, and that's what makes it very, very complex and difficult to address. But there's no doubt in my mind that a number of these factors up here is influencing the immune function and changing the risk of infectious diseases in the animals. This is just a slide to underpin the complexity of the immune system. Major challenges or changes are occurring around calving, homoerotic regulation taking place, changing a number of the hormones, and that is then also triggering changes in some of the metabolites and nutrients on this side, including also some of the immune proteins. There's, of course, here in this respect, a tight link to the peripheral tissue, mobilization of body tissue, and the yield. But the liver also plays an important role in all these aspects, and we may have fatty infiltration. We may have reduction in some important immune proteins, including the acute phase proteins, and the situation may impact the synthesis also of other important proteins in the animal. But we have opportunity to change nutrition, for instance. So I'm only digging into one small part of the situation, and also here giving a few examples on glucose and glutamine supply. Why is that important? It's a nutrient for the immune cells. It's primarily using glucose and glutamine as energy source. But glutamine constitutes normally 43% of the amino acids in muscle tissue in dry cows, but in early lactation it's changed to only 24%. So potentially we may have nutrients here that are lacking for immune cells. We also know that when the cow is having trouble, or any other animal has trouble, the glutamine concentration in plasma may drop 50%. So that's quite a bit. An interesting thing about the immune cells is also that some of them have a lifespan that's very short. And if they are generated in the period where the cow has very low concentrations of, for instance, glucose or some other important nutrients, that may impact the effect of these cells when the animal is challenged. I will not go into very many details here because I'll refer you to a review that we have done, but just show that glutamine and glucose are two nutrients that has only stimulatory effects. It is involved in, or it's stimulating cytokine production, also reactive oxygen, it should be, metabolite production, cell division, phagocytosis, etc. But don't get confused. The situation of cytokine production may not always be beneficial. You may also have situation in early lactation, for instance, TNF-alpha overproduction that actually is killing cows. So it's just to mention that it's complex. Similar for glucose, many important aspects. For ketone bodies, we see the opposite. We actually only have inhibitory factors, and that is on a number of parameters, including the important phagocytosis and oxidative burst that's important in leukocytes. Fatty acids, there we see both inhibitory and stimulatory effects. Again, I won't go into details, but it has to do with the complexity of fatty acids. We have long and short chain fatty acids. We have saturated, we have unsaturated and polyunsaturated fatty acids, and they have, as far as we know, specific effects on some aspects of the immune system. We still have, in my opinion, a lot to learn about this situation. Energy balance, similar, but again, I think that's linked to some of, I think that's linked to some of the former metabolites that's very closely correlated to energy balance. Now, I mentioned that energy balance was difficult to measure, and then I showed some blood parameters. Not very many farmers go around taking blood samples for getting the status of the animal. So actually, for the last 10 years, we've also been working on trying to get information from readily accessible fluids, such as milk. And we've been involved in developing the herd navigator. That's a system, that's one of the industries from Foss Electric and De La Valle that had made a joint venture on a system that automatically takes a blood sample and measures ketone bodies, LDH for assessing mastitis, but also progesterone in order to get the reproductive status of the animal. And we have been keen on also looking further into if we could find parameters in milk that could be linked to physiological imbalance, because we do not think that beta-hydroxybutyrate necessarily is the best in that respect. So here is, maybe I should explain what we did. We had 29 healthy Holstein cows, some in early lactation, some in mid-lactation, and then we had a pre-period and a period where we added 60% of straw to their normal diet, but gave it ad lib. But basically, the cows had 40% of their net energy for lactation, so it was kind of to resemble an off-feed challenge situation. And here's what we found: a number of parameters that is changed markedly, and we are currently working further on isocitrate, because we can see that within short time we can actually pick that up in milk. And basically isocitrate, but maybe just as important, free glucose, because free glucose is dropping. At the same time, milk yield is dropping, so if you take the multiplum of those, you have a marker that's just as strong as this, or even better. So we are further investigating how that is, how that situation is. But look at, for instance, some beta-hydroxybutyrate, as I mentioned, it's in here. It's not really kicking out significantly, and we're using beta-hydroxybutyrate as one of those in early lactation that we trust telling us about energy balance. We also did it in late lactation, and the changes are just a little less significant, but actually it's the same picture. So have we solved the problem? No. There's quite a bit of work yet. And I think we need to look at individual differences. These are kind of obvious, but what is going on in the cows is less obvious. We need to understand these individual differences. We need to be better in describing cow status and risk. We need to work on animal status, strategies, and risk management. And at least for dairy cows, probably also for sows, it would be possible to do that on an individual basis. This is basically just telling the same, so I'll go to the last slide here. I think for a number of years we'll still have to accept that we get more knowledge, but basically there's a lot of black box situations yet, also in these production animals. But we will be working on the inputs, and we'll be looking on risk of diseases. We will be looking on risk of suboptimal performance production. We'll have to come up with some common currencies, because we can't measure all the factors in the immune system, or for that matter any of the other systems. So we will have to find some common currencies that gives us good signals on animal status. For that, we need to search for indicators. That could be in the rumen. It could be on nutrients, metabolites, hormones. It could also be on some of the production profiles where we, for instance, looking at acceleration in milk yield. But the key thing is we need to have input primarily automatically, because of the number of animals that's available on farms nowadays. And we need to have systems that can generate the philosophy or the risk and feed back into the situation for the individual cow. We have that technology, but we need more science, more combinations, I think, of biology and technology in order to have a situation where we improve on health and welfare, and at the same time we have optimal production. Thank you.